Welcome viewers. Today we are going to talk about the different antibiotic resistance mechanisms of bacteria and how we can study the different antibiotic resistance mechanism of bacteria experimentally. Now before we start off with the experimental procedures, we should know what are antibiotics. Antibiotics are secondary metabolites of microorganisms which are generally used to kill the other group of bacteria, or they can be used to prevent the bacterial growth. The word antibiotic is derived from the word antibiosis, which is a term coined in the year of 1889 by Louis Pasteur's student Paul Willemin. It means a process in which life is used to kill another life. Now, before we go into greater details of antibiotics, we should know how can we classify the antibiotics. Antibiotics can be classified on various ways. One of the way is it can be classified on its mode of action, that is how it is killing the bacteria. It can be used on the mode of action. Antibiotics are bactericidal or bacteriostatic in nature. Cidal means the antibiotics are completely killing the bacteria, while static means it prevents the bacterial growth. The other way of classification or classifying antibiotics is through its route of administration. That is, either it is injected or taken orally. Likewise, if taken orally, it is assimilated into the system through the digestive tract. If injected, it is directly administered or introduced into the circulatory system. Again, on the type of bacteria the antibiotic kills, it can be divided into narrow range antibiotics, like the antibiotics are specific in its action against the gram-positive or gram-negative bacteria. Again, the wide range antibiotics which are generally used to kill a wide number of bacteria in lab. The first discovery made in the field of antibiotics is by Sir Alexander Fleming. He was a Scottish biologist, and he saw that when he was treating patients of World War I, some of the patients died due to severe bacterial infections. It was then during this period he discovered the important antibiotic penicillin, which could save many people's life. He found that when he grew Staphylococcus aureus on a Petri plate and left it for some days in his lab, another contamination he found in the Petri plates. It was a contamination made by the mold Penicillium notatum. After close examination of Penicillium notatum on the culture plates of Staphylococcus aureus, he found that the colonies of Staphylococcus aureus in close vicinity to Penicillium notatum were inhibited. On further examination of Penicillium notatum, he found that Penicillium notatum secreted the secondary metabolite compound penicillin, which acted against the colonies of Staphylococcus aureus. So with time immemorial, several other antibiotics were discovered from the microorganisms. Like Streptomycin was discovered from Streptomyces, then nystatin was discovered, which is used commonly for fungal infections. In this way, now we have the diverse range of antibiotics discovered from time to time. Now let us move to the pathogenic microorganisms. Before we move, can you name me one pathogenic microorganism?
E. coli.
E. coli. E. coli, the full name of E. coli
Escherichia coli.
Escherichia coli. What kind of infection does it cause?
UTI, urinary tract infection in us.
In us, very good. The pathogenic organisms can be divided into two main types, aerobic and anaerobic. Can you name me one anaerobic pathogenic microorganism? Anaerobic pathogenic microorganism and the disease it causes.
Clostridium.
Clostridium. The species
Clostridium tetani.
Tetani. It causes tetanus. Very good. So, if we see the list of aerobic pathogenic microorganism, so what does it show? It contains the important microbes like Staphylococcus aureus, Streptococcus pneumoniae, Klebsiella pneumoniae, then Escherichia coli, Pseudomonas aeruginosa, Haemophilus influenzae. So, all these aerobic organisms are present everywhere around us, even in the air present in this room. And so, the antibiotics are able to treat the infections caused by these microorganisms in us. Similarly, we have the anaerobic pathogenic microorganisms like Clostridium tetani, Peptococcus, etc. We have botulism disease also caused by Clostridium botulinum. Very good. So, in this way, the antibiotics are used to cure a wide range of diseases. In the earlier sections, we had talked about how we can classify antibiotics. One of them is through mode of its action. Each antibiotic is able to kill bacteria through its specific mode of action. We know that the bacteria can be divided into two main types. One is gram-positive bacteria, another is gram-negative bacteria. Now, gram-negative bacteria, they have a membrane which is absent in gram-positive bacteria. So, the antibiotics which target the cell wall, they are cell wall inhibiting antibiotics, and they generally kill the growing vegetative cells because cell wall synthesis takes place in this kind of bacteria. We have antibiotics which target the bacterial cell membrane, and hence in this way they kill the bacteria. We have antibiotics that target the DNA or RNA synthesizing machinery and impair the synthesis of DNA and RNA. We have antibiotics which attack the cytoplasm and inhibit the cytosolic enzymes. Some of the antibiotics which are able to penetrate the cell wall or inhibit the cell wall formation are penicillins, cephalosporins, cycloserines. How they inhibit the cell wall formation, these antibiotics?
By preventing the formation of peptidoglycan, the cross-linkage of peptidoglycan.
Very good. Peptidoglycan layer, which is a component of the cell wall. So, these antibiotics, they degrade the peptidoglycan layer, and in this way, the cell wall synthesis of growing bacteria is prevented. We have antibiotics like polymyxins, which attack the cell membrane. Very good. So, they attack the cell membrane and they inhibit the formation of cell membrane. We have sulfonamides which attack the cytosolic enzymes and inhibit the synthesis of folic. And folic is an important constituent or component of nucleotides. And nucleotides is required for DNA synthesis. Again, we have antibiotics like aminoglycosides, tetracycline, chloramphenicol, which can inhibit the RNA synthesizing machinery by protein synthesizing apparatus. Like it binds with the ribosomes and it impairs the synthesis of proteins. We have rifampicins or rifamycins which bind with the DNA dependent RNA polymerase and it impairs or prevents the synthesis of RNA. Chemotherapeutic index is defined as toxic dose by therapeutic dose. It is generally associated with an antibiotic. It is the highest concentration of the drug that can be tolerated by the host to the lowest concentration of the drug which will kill the infection agent. There are several mechanisms of antibiotic resistance of bacteria. Like if the antibiotic is specific in its mode of action, like it inhibits the cell wall synthesis as we have seen with the penicillins. How it can avoid the antibiotic? By altered cell wall composition or by the destruction by beta-lactamases. Beta-lactamases are your compounds which destroy the beta-lactam ring found in penicillin or in cell wall attacking antibiotics. Then we have the cell membrane antibiotics. Bacteria can develop resistance against them by altered membrane structure. Then the antibiotics which attack the cell membrane will not be able to degrade the cell membrane.
Ma'am, how does bacteria get resistant to polymyxins?
Now the polymyxins attacks the cell membrane of bacteria. Now the bacteria can develop resistance against polymyxins by altered membrane structure. If the membrane composition or the chemical composition of membrane is altered, then polymyxins won't be able to bind with the cell membrane. Similarly, we have inhibition of DNA synthesis, like the fluoroquinolones which bind with the DNA, so the microbes can develop resistance against fluoroquinolones by inactivation of drug or by altered drug target. Similarly, we have your inhibition of RNA synthesis and rifampicin or rifamycin. It binds with the DNA dependent RNA polymerase and prevents the RNA synthesis. But when the bacteria develops resistance to rifampicin by altered drug target, then your rifampicin is not able to bind with the RNA polymerase enzyme. In this way, the microbes, they have developed several mechanisms through which they can evade the antimicrobial effect or the antibiotic effect. And these group of bacteria are termed as antibiotic resistant bacteria. And they are the very dangerous group of bacteria because we cannot kill them by any antibiotics. The first among them is Kirby-Bauer test procedure. In this procedure, we take a Petri plate like we have it here, which is filled with media. You can see to a depth of or to a height of 5 millimeter. Now the media which is used in case of Kirby-Bauer test procedure is Muller-Hinton agar. Muller-Hinton agar produces satisfactory growth of all pathogens which we use for studying the antimicrobial resistance. Then it is very low in sulfonamides, trimethoprimines, tetracycline inhibitors, and it produces reproducible results. We take disc or filter paper of uniform size absorbed with different concentrations and placed on the surface of the medium. Now, when the bacteria is resistant to the antibiotic, it will produce no zone of inhibition. But if the bacteria is susceptible to antibiotic, it will produce a zone of inhibition. Based on the diameter of the zone of inhibition, we can say that the bacteria is either susceptible, resistant or intermediate to the antibiotic.
Ma'am, what is secondary zone of inhibition?
Apart from the primary zone of inhibition, a secondary zone of inhibition is formed. Now, in this zone of inhibition, there are bacteria which are resistant to that group of antibiotic, and hence it produces 1 or 2 colonies in the secondary zone of inhibition. Another main cause of secondary zone of inhibition is that the bacteria which we are using is not a pure culture. It contains mixed population of bacteria. So some of the bacteria might be resistant to the antibiotic and hence it will grow in this secondary zone, and accordingly it is known as secondary zone of inhibition. We require Petri plates with solidified Muller-Hinton agar media. Apart from this, we require a pure culture of bacteria. The pure culture of bacteria or the optical density of pure culture of bacteria has to be brought down to a uniform density. According to the NCCLS, that is National Committee on Clinical Lab Standards, they have suggested that a 0.5 McFarland standard bacterial culture should be used. The bacterial culture should be like that of Staphylococcus aureus or your Escherichia coli, against which we want to find the antibiotic resistance. We require antibiotics, standard antibiotics, whose concentration we know, like streptomycin, tetracycline, penicillin, it can be anything. We first take a Petri plate like I have it over here, and then with the help of a antibiotic disc dispenser, we dispense the antibiotics on the surface of the agar plate. Now before we dispense of the antibiotics on the surface of the agar plate, we first need to inoculate the bacteria, the pure culture of bacteria, against which we want to perform the antibiotic resistance mechanism. We spread the bacterial inoculum of the pure culture on the surface of the agar plate. After the bacterial inoculum has got penetrated into the agar surface, then we dispense the antibiotics with the help of antibiotic disc dispenser or with the help of a sterile forceps. This is the antibiotic disc we can see of different colors. It represents different kinds of antibiotics. Then it is placed on the agar surface with the help of sterile forceps, and each antibiotic disc should be at equidistance from each other. Now, the antibiotic discs are placed in such a way that it adheres, not presses too deep down on the agar surface, but just adheres onto the agar surface. Then after incubation in a incubator, 37-degree incubator, for 24 to 48 hours, we observe the zone of inhibition. We measure the zone of inhibition or the diameter of zone of inhibition with a millimeter scale. Based on the diameter of zone of inhibition, we can say that the bacteria is either resistant or susceptible or intermediate to that particular group of antibiotic. So in this way, by using a single strain of bacterium, we can assess the different antibiotic resistance of bacteria. So in this case, the bacteria is a pure culture, but we are using different antibiotics to assess the antibiotic resistance of bacteria. Suppose this is a Petri plate which is showing confluent bacterial growth on its surface. Now, when the discs are placed, we can see that there are 5 different antibiotic discs. After 24 to 48 hours of incubation, we can find that there are zone of inhibition formed. Now, based on the diameter of zone of inhibition, we can say that the antibiotic is either resistant or you are susceptible to that particular antibiotic. For example, in this case, suppose this is a disc A. You can see a greater diameter of zone of inhibition. So we can say that in this case, this disc A or the bacterium is susceptible to this particular antibiotic. Again, we have a smaller zone of inhibition here. Suppose this is the antibiotic C. So the diameter of the zone of inhibition is less here. So this antibiotic is less susceptible. So it is developing the resistance mechanisms. Again, if you see this antibiotic disc, it is resistant, or the bacteria is resistant to this antibiotic as there is no zone of inhibition. Very good. Again, in this case, you can find that this antibiotic has produced little zone of inhibition. So in this case, we can say that the bacteria is developing resistance mechanism to this particular antibiotic. So in this way, we can study the different resistance mechanism of a particular bacterium to different antibiotics. Similarly, the same figure is represented here. In this case, you can see there are multiple discs placed, and all this disc represents antibiotics of different types. Now, some of the antibiotics or the bacterium is resistant to some of the antibiotics, while it is susceptible to some of the other kinds of antibiotics. In this way, we can find out the range of antibiotics to which the bacterium is either susceptible or resistant. This assay is also known as disk-diffusion assay.
Ma'am, can the use of multiple drugs at the same time affect the efficiency of antibiotics?
Yes, it can affect the efficiency of antibiotics. When we are using more than one antibiotic, then the process is known as combinatorial drug therapy. Now, the two discs which are placed on the agar surface might have an additive effect on the bacteria, or it might have a synergistic effect in killing the bacteria. Synergistic effect means it is producing a greater zone of inhibition when the two antibiotics are used together, and thus the efficiency of antibiotic it increases. It depends upon the type of antibiotic and mode of action on the bacteria. The precautions which we need to follow during Kirby-Bauer test procedure is the inoculum density of the pure culture of bacteria through which we are going to use for seeding of the bacterial plates should be adjusted or maintained according to NCCLS standards, that is 0.5 McFarland standards. Another point, very important point, is the antibiotic disc when placed on the agar plates should not be pressed deep down on the agar surface. It should only adhere onto the bacterial agar surface. The strain which we use for studying the antimicrobial resistance should be of pure culture. Otherwise, it might lead to the formation of secondary zone of inhibition, or it might produce your confusing results. So these are the certain precautions which have to be kept in mind during performance or performing the Kirby-Bauer antibiotic test procedure. The other method for determining the antimicrobial resistance of bacteria is MIC. MIC stands for minimum inhibitory concentration, and it is also known as broth dilution method. We require a nutrient broth solution which are dispensed in sterile test tubes. We require a pure culture, that is Staphylococcus aureus, a standard concentration of antibiotic. We require the sterile 2 ml and 10 ml pipettes. We require spectrophotometer, glassware marking pencils, spirit lamp, etc.
Ma'am, what is the purpose of Erlenmeyer flask in the MIC experiment?
Erlenmeyer flask is the common flask which we use for growing bacterial culture in lab. It contains the medium, and when inoculated with the bacterial strain, it is placed on a shaker. Now it allows the growth of bacteria in a larger volume. So this is used as a source of the bacterial strain, which is used for performing the MIC. We first take the 10 tubes labeled with the help of glassware marking pencil, then nutrient solution which we add 2 ml to each of the tubes. Now in this case, if you see that tube 1 is acting as a negative control, because it contains only the antibiotic and the bacterial strain. The other tube 10 is a positive control, because in this tube the antibiotic is absent. Now, the antibiotic is serially diluted with the help of broth. Take 2 ml of nutrient broth medium in sterile tube, that is number 2, and then you add penicillin 2 ml. Now from this, so it totals 4 ml. You again take up 2 ml and transfer to the tube number 3. In this way, you go for the serial dilution till tube number 9. Afterwards, from tube number 9, you discard the extra 2 ml. So, the total volume till now from 1 to 9 is 2 ml. Now, you add a test culture, that is 1:1000 times dilution of Staphylococcus aureus to each of the tubes. That is, each of the tubes you are adding 2 ml. So the total volume becomes 4 ml. This is your antibiotic stock. So you know the concentration of antibiotics since you are using pure antibiotic solution. Now when you are diluting it, the concentration of antibiotic is reduced. So that's why now you can find out at which concentration the growth of bacteria is completely inhibited. So that's why it is known as MIC, minimum inhibitory concentration. If you look at the result, you can see that there is growth only in the first 3 test tubes or the culture tubes, while in the rest of the culture tubes there is no growth. Because the antibiotics here, it is in low dose, while the antibiotic present in the 4th is able to inhibit the growth of bacteria. So in this way we can find the MIC of a particular antibiotic against a particular strain of bacteria. Now similarly you have tetracycline. The tetracycline, again in this case you can see the concentration. The concentration of 1.5 is completely able to inhibit the growth of bacteria. So in this way the MIC is helpful in understanding the minimum concentration of your antibiotic which the bacteria is not able to prevent, or the highest concentration of antibiotic which the bacteria can resist. First, the concentration of the antibiotics should be carefully diluted. The culture which we used for MIC should be of a pure culture, and there should be no mistake in the serial dilution step. And after the addition of antibiotic and your culture, it should be mixed well. Now let us conclude of what we have learned today. We have learned that antibiotics are very important compounds or chemicals used to kill the different groups of bacteria. Now the experiments which we perform in lab for assessing the antimicrobial resistance is either a disk-diffusion assay, commonly known as Kirby-Bauer test, or we have the broth dilution assay, commonly known as minimum inhibitory concentration. After knowing that a particular bacteria is susceptible to a particular antibiotic, we perform the MIC or minimum inhibitory concentration. With the help of MIC assay, we can determine the minimum dose which can be used to kill the bacteria and prescribe that dose for human beings.